first of all, massive welcome to everybody. It's a real pleasure to have everyone here for the Coffee Buddies inaugural XY Factor competition, and we'll talk a little bit more about that as we go along. But first of all, welcome to Coffee Buddies and at the live at Biotechgate Digital, which is Patrick's great invention, and we're so proud and pleased to be part of a festival of meeting people. And we've this is our number, I think this is our number 6, is it, Graham? 5 or 6 that we've done already. And our Coffee Buddies initiative is really we ramble around certain areas and we bring in guests, and our raison d'etre, with a big R, if that's how you spell it, is to entertain, engage and inform. And these tend to be cameras on events where we involve people, so people get a chance to hear their voice spoken. And somebody once said to me that nobody really arrives until they hear their own voice. So I think that's something that we can all relate to. So this is a fantastic time, and this is our inaugural XY Factor. And why have we called it the XY Factor competition? Well, somebody already got the idea of an X Factor, so we couldn't get that one. So we thought we'd add the Y to it, because it's sort of, we're all into the interest of Y. And what's the goal? The goal really is to bring interesting companies together in a different format to the normal format that we're used to, which is pitch presentations, which are okay, but sometimes they can be a little bit dry and non-interactive. So we're doing pitch interviews, which is always my pleasure. And we're also delighted to have our sponsors for the event, which is MSD. So thank you for them. And the format is that we've got a group of people who will be judging. And firstly, our judge of the event is Tom, which maybe, Tom, you could just say hi to the group. Oh, Tom, you're on mute, my friend, or something.
Yeah.
Just say hi.
Hi to group.
And Tom, just what do you do, just so that people get a little bit about your background?
Yeah, I basically help a lot of young companies, both for fees and mainly pro bono. But I've been director of neuroscience, head of biotech companies in the US. Been involved with blockbuster drugs over the years, so I think I know a little bit about drug discovery and development, but just a little bit.
Fantastic. Well, welcome, Tom, and we're delighted to have you. And then the other member of our panel is Christine. Where's Christine? I can't see. Oh, there you are, Christine. Christine. I mean, Christine, you're from MRL Ventures, which I know very well. Maybe you could just tell everyone who you are and what you do.
Sure. So Christine Brennan, I'm a partner at Emerald Ventures Fund, which is the strategic corporate venture arm of MSD. I'm physically in Cambridge, Massachusetts this morning, also a PhD and postdoc in neuroscience. So Tom, you and I should catch up. And have spent most of my career in biotech and big pharma and business development before jumping into ventures. And then I've been in ventures now for about 8 years at Novartis Venture Fund, and now here at Emerald Ventures Fund.
Fantastic. And we've got another colleague joining us, but I don't know whether they have yet.
Yeah, but they haven't joined us yet, but we have got Jia here. Hi, Jia.
Great. Where's
Would you like to say hi to everybody? Introduce yourself.
Hi, everybody. My name is Jia. I'm the managing director at Biotechgate Capital, which is a French private equity fund. I'm part of the new Cathay Health Fund, which invests at the convergence of healthcare, tech, and life sciences. My own background is in genetics. I did research at MIT for a number of years. Before going into the investment world, I was part of Partners Group, and then later spent time at Philips Healthcare, as well as worked on a few startups. Right now, I'm based in London. Nice to meet everybody.
Jay, thank you so much for coming, and thank you to the panel for today. So we're going to go now through a number of companies. I think, Graham, we've got 5, although Graham said we had 6 this morning, but we've got 5, and we're going to go through. It's 10 minutes of a conversation with 5 minutes questions, which we will ask our judging panel, which is Tom, and we've got YE, and we've got Christine, who can ask any questions, and then we will move on to the other one. So it's only going to be high level stuff, but if you have got some slides, maybe you could put them on and do it quickly. So let me just start with Jeanette. She's the first of our XY Factor contestants, and welcome to Jeanette. Maybe you can just tell everyone who you are and what you do.
Yes, hello, I'm Jeanette Thomas, and I've founded a company with 3 other people called Pellis Care, and we're developing a treatment for chronic wounds like venous leg ulcers and diabetic foot ulcers.
Fantastic. And maybe you could just tell us why you've focused on chronic wounds.
Yeah, it's a really huge unmet need. It's a $100 billion global market and around 100 million people affected worldwide. Venous leg ulcers, 21 million people have chronic venous leg ulcers, and their wounds are not healing, and they suffer terribly over several months, or if not years. And we all know people that have had their legs constantly bandaged, and it's a very debilitating disease. And we have a treatment that we think will heal it, and we have seen evidence in people where their wounds have healed. And I could, if you could bear it, show you a photograph quickly of a wound that's healed.
Well, that's not very pleasant, so we'll keep that off. we'll keep that off. But I mean, you know, it's a massive unmet medical need. We know that. So have you got a drug? Is it an ointment? Is it a drug? Yeah. What is it?
It's a cream, and we've just had a GB patent patented, and we have international filings for a therapeutic cream containing a new use for an active ingredient that has been used in other things. So we've discovered that it's got a lot of anti-inflammatory properties, and it is those properties that are used to treat the chronic wounds and that show healing within 12, 13 weeks of treatment.
And can you reveal the active ingredient? Or
yeah, it's patented. So it's a triclosan in the cream at higher dose than usual.
Okay, and I mean, so your idea is that you're gonna use this cream or ointment. I mean, what's if you look forward to the next 3 months or 6 months, what's the value inflection you're after? Where are you looking to go?
Well, we've actually had meetings with the MHRA, scientific advice meetings, to confirm we don't need to do any further research work before doing a clinical trial. So we're raising 2.5 million in order to do the manufacturing process development so that we can do a clinical trial in 45 patients to show that it really does have efficacy, and that will be the main value inflection point. And that trial will take approximately a year.
And what's the standard of care now?
The standard of care for VLU is compression bandaging, and patients have compression bandaging changed every couple of weeks. So they have to go to specialist nurses or podiatrists, depending on the country, but specialist care providers to do that compression bandaging is very onerous cost-wise for healthcare provision, and also for the patient, it's not a pleasant experience.
But would you not imagine that they will have the bandaging and the cream? Won't that be the way it will be used?
It will, but then it's a chronic wound at the moment, so that carries on for months or years, whereas if they heal, then they won't need it once they've healed, they will no longer need the bandaging.
Yeah, and I mean, you're thinking of your value inflection, or your inflection point is a clinical trial on 45 patients, with your control arm being what? A placebo cream. And so they're the standard of care, so the patients get treated as well as they do now. There are no other treatments. They continue just compression bandaging. So they'll have standard of care plus our cream as another arm. And then after that, where would you go? Where will you go after that?
Yeah, so after that, we will do a phase 2B study. So the phase 2A is a safety and evidence of efficacy. And then we'll continue the development program as you normally would, which would be a phase 2B leading into a phase 3 study. And we would also like to bring in other indications. There's a really huge unmet need in diabetic foot ulcers.
I'm getting a wave, which means the time is so...
The little wave was just that Michael Solako's joined us.
Oh, okay,
so,
that's not the time.
Good to see you here, Michael.
But how are we doing for time, Graham? It wasn't a wave on the time.
We've got 5 minutes still.
Oh, we've still got 5 minutes. 5 minutes, including questions.
Oh, Yeah, 5 minutes including questions. Yeah.
Okay, so my last question then to you, Jeanette, where are you? Oh yes, Jeanette. What are you looking for from an investor or from external parties? What are you after?
Yeah, we want to raise £2.5 million pounds in order to develop the manufacturing process to GMP and perform the clinical trial.
And is that going to be tranched? Do you want it tranched, or do you want it in one go?
Well, in one go, essentially, or the promise in one go, because there's no real value inflection or change until we get to complete the clinical trial.
And my final question, what's the exit strategy?
Yeah, the exit strategy will be to license or some sort of M&A at the end of the phase 2 clinical trial. But we recognise that there's actually great opportunity for further IP from the clinical trial, so we may continue on. But I think that if we can show efficacy, there'll be great value in that project.
And timeline for that exit?
2 to 3 years.
Fantastic. Well, thanks, Jeanette. I'm going to ask the group. We've got Tom, Christine, and who's joined us? Graham, did you say?
Michael Solako is there.
Sorry, hi, Michael.
He obviously
didn't hear it. So I'll ask Tom and Christine to ask. Christine, did you do some waving at me? Yes.
So these are always a tougher case for venture investors to invest in, mainly because the IP is usually short-lived and based on method of use. So just wondering, you just mentioned maybe having a little bit more IP based on the clinical trial. I'm sure IP based on formulation, but just would love to understand kind of your IP strategy and how to make this exciting and a patent length that makes sense.
Yeah, so we've just got a GB patent. So we have a 20-year patent for, well, now 18 years for a use patent. And it combines the use with the formulation, so you can't. We think we understand that we'll have potentials for various different mechanisms of action, and if we can understand the mechanism of action through the clinical study, that might lead to other indications as well. And I mean, you never know what's going to come up when you do the studies, but we'll be looking at biomarkers through that clinical study.
Tom.
Yeah, absolutely fascinating area, Jeanette, particularly, in the diabetic. With regard to funding, have you approached any of these patient groups or charities?
Not yet, but I think that's a good idea. We've talked to a lot of patients and understand how badly they suffer. But I think that is a good way to go. We're looking also, we can leverage with grants once we get some initial investment. Yeah,
see, I see that as a very important, approach for you, in the sense that you get the patients on board.
Yeah. And,
particularly word of mouth, would then help you, in that process. So I'd advise you to go, that route as quickly as possible.
Yeah, we are also in conversation with Singapore Wound Innovation Centre and Professor Keith Harding at the Welsh Wound Innovation Centre, and they've got direct patient contact. But I do take your point there.
Fantastic. Graham, how are we doing for time?
Yeah, we're doing good for time. I know Michael was busy,
so
out
Christine,
a lot, but do you have a question on what you heard, Michael?
None, I'd say now, but sort of apologies, Jeanette, I missed some of it, but yeah, it sounds interesting.
We'll get you connected after.
Christine's got one other thing she'd like to say. Christine?
I do, thank you. So it's such a huge unmet need. I'm also just wondering, the $2.5 million gets you through, it sounded like phase 2A.
Yeah.
What do you think you'll need to get through to that phase 2? It sounded like after phase 2B, you thought you might be at a point where it could be more attractive from a licensing M&A standpoint.
Yeah.
So just wondering what the full boat of money that you'll need to get to that inflection point.
Yeah, so I think it'll need for the phase 2B would be another 15 million, and we can start looking at the diabetic foot ulcer in a small study as well. Yeah.
Well, I think that was fantastic. Jeanette, awesome stuff. Jeanette, how can people contact you?
Yeah, jthomas@pelliscare.com. I'm on LinkedIn, and I will put a note here in the chat.
Well, I think, that was a great start to the XY competition. Thank you very much. And we'll give you a round of traditional E applause, which I don't know what the E stands for, but that's what it is. So we're now going to move on to the next one. Now I'm just looking around for Ross. Is Ross in the room? Where's Ross?
Where is Ross? That's a good question.
Well, there he is. He's down in the corner. I found him. My goodness, Ross, you're in the corner. Ross, welcome to the XY Factor. Ross, maybe you could introduce yourself. It's a delight to see you, my friend.
Sure. My name is Ross Breckenridge. I'm the CEO of Arjuna Therapeutics. And as you can tell from my broad Spanish accent, we're a Spanish company. We're a spin out from the University of Santiago de Compostela. And we are developing for medical use a completely new class of small molecules that were discovered, or the manufacture of them was discovered at the University of Santiago. So our molecules are very small, unique. They are now easy to manufacture, that somebody has spent 10 years figuring out how to manufacture them. We have quite substantial patent barriers, and our lead molecule we're being very ambitious about. It's a molecule that specifically targets cancer cells that have dysregulated energy metabolism. And it turns out this group of cancers is enormous and untreatable. It includes cancers driven by mutations in KRAS and also glioblastoma. So it includes the really big, nasty cancers. And we think that AG5, our lead molecule, will tackle this new class of cancers.
So tell us a bit, what is AG5 and how long has it been hanging around?
Well, in terms of manufacturing patents, the manufacturing patents were started to be filed about 4 years ago, and those patents are continuing. Actually, the more people you speak to, the more people I speak to, people have been trying to make these molecules for years, but they do them in femtogram quantities by doing crazy things like heating metals in a vacuum. And then you get a femtogram that you have your entire PhD to work on. We're working on ways to make 100 milligram quantities plus and in solution. So these are tiny uncharged molecules that will penetrate throughout the body. We've done a lot of work on PK and tox, and essentially it reduces the body to a one compartment model. So it'll freely cross across the blood-brain barrier and into tumors that have no vasculature.
So just tell me again, what's in the inventive box? What's the, if you like, the magical thing that you own?
Okay, so remember from O-level chemistry, the idea of a metal is a sort of a crystal of metal nuclei with shared outer shell electrons. What our technology is essentially imparting a huge amount of energy into metal atoms when they're in a soluble ionized form, which persuades the outer D shell electrons to become excited and then form covalent bonds with each other. So essentially they're covalently bonded metal atoms. Once they're covalently bonded, the only thing that we found that can break them up is actually a synchrotron radiation beam. So they're incredibly stable, they're not metabolized, and because they're so small, they will passively diffuse into mitochondria, into cells and across the blood-brain barrier.
So your invention is that you can create these molecules in a way that no one else has been able to do before.
Yeah, that's right. In ways that make them actually practically useful.
And what do they do biologically?
Well, it's fascinating. Each molecule in our pipeline has a completely different mechanism of action. So our first molecule, AG5, is actually a catalyst. So when it goes into most cells, it doesn't really do anything. But if it encounters very high levels of superoxide, which is one of the reactive oxygen species, it binds it and stops it decaying, and then catalyzes oxidation of sulfur atoms. And the only place that superoxide is really present in the body is the mitochondrion. And the specific amino acid motif that AG5 oxidizes is present in antioxidant protein molecules. And here's a really interesting bit of evolutionary biology. Obviously, mitochondria used to be, in the past, bacteria. And bacteria have a very primitive antioxidant system that has developed as they've become endocytosed into the cell, into two redundant pathways, two redundant protein antioxidant pathways. The idea of killing cancer cells by knocking out the brakes, if you like, knocking out the antioxidant molecules is not a new one, but it's always floundered because there are two pathways that are redundant. So if you knock one out, the other one will take up the strain and the cell will survive. If you knock them both out pharmacologically, then you also kill the cells in the heart and in the brain, which in general we don't want to kill. So our approach, by using a molecule that's only activated in the cell that you want to kill, and then knocking out both pathways, we think actually circumvents a lot of the problems.
You've got a pattern on a method of creation, but then each one of these molecules works in a different mechanism of action. So do you then have a patent on the individual molecule on a mechanism of action?
So they're all manufactured by separate processes, which are all patentable.
So is this a bit like when we used to, there was this whole world of silicon chemistry where you created a whole new molecular class? Is that what this really is?
Yeah, it is. It is a new class of molecules. So these haven't gone into man.
And you can do, how do you bioassay them? How do you know how much the plasma concentration is?
Yeah, so we asked the FDA about this, and they said, why do you just do the same thing that you do for oxaliplatin, which is just measure the amount of metal atoms? And as long as your patient hasn't worked down a silver mine or been making silver cutlery, the atoms, the silver atoms that you're giving, are the silver atoms that you're measuring in the blood are the ones that you've given. And it turns out that oxaliplatin, that's exactly the same way they measured oxaliplatin levels, just measuring the platinum atoms.
So can you go through a classical route of an in vitro then an in vivo experiment? You can do a classical, you can get a functional readout. Is that what, so tell us about, how would you functionally read out one of these? You know, I've got the molecule, I've got a cell, what do I measure?
Well, we know we've got a very fine-grained view of what the mechanism of action is. So we can actually measure target engagement. We can measure directly measure the oxidation of the antioxidant proteins. There are very good antibodies for this. So we can measure that in vitro. We can measure that in biopsies, in cells, in animal models. We obviously haven't gone into humans yet, but we're certainly intending to do that. But one of the big things that we're looking at is actually how we look at biomarkers, because of course you can't biopsy tumors immediately after you've given your drug, but you can take blood test results. So we've done a fair amount of work on biomarkers in cells and in animal models, and we've just started a collaboration with Deloitte actually, who are working with us to embed machine learning into our clinical program so that we can sequentially train a data set to understand more and more about what our molecule is doing before the really big financially risky experiment, which is of course the phase 3.
I've got loads of questions, but in the spirit of timing.
You've got a few minutes, Tony.
Yeah, so I mean, you know, the bigger picture, Ross, we go to the bigger picture. So you've got a moiety, a chemical moiety, which you can understand. What's the pathway forward, and where's your next value inflection?
Well, we think that actually we're going to be able to justify doing a healthy volunteer study as our first in man. So we're aiming for an IND in 12 months, and we're going to do a small PK/PD study in man that will enable us to a lot more rapidly progress through our dose escalation in patients. We're aiming to interrogate the mechanism of action in 3 separate disease-specific groups, which we've identified biomarkers in. And then we're going to go on after, and that basically is what our Series A will pay to get to the end of that. And the next raise after that will be to pay for the registration trial. And what we will do is initially we will pick one indication to do a registration trial in, and then after that we're going to if the results are what we hope, we're going to do a tumor agnostic trial based on biomarkers across solid tumors. You know, we think that this will treat 20% of all cancers.
So what's the one or two compelling arguments that you would throw out to the investors to come and give you some cash?
Well, I mean, this is not the 75th PD-L1 inhibitor. This is something that's entirely new, and we're actually aiming for a group of cancers that really don't have any incumbent therapies. So, you know, for any new chemical moiety that has a completely new mechanism of action, of course, the risk is higher. But then we think that the potential upside is enormous. The other thing is that this is a pipeline. There are a whole lot of other indications that are less risky to do with this molecule and our follow-on molecule, and also non-cancer indications as well that are patented. So this is this is going to be a new class of medicine, and you know, it doesn't have to win in KRAS cancer for it to be an extremely large return on investment from an investment at this stage.
Well, Ross, can I thank you massively for a fantastic delivery? Lots of questions, but let's pass it on to Tom, Michael, and Christine for some questions to you, Ross. Who would like to start?
Christine.
Christine.
Sure. So first, how much money are you raising? I know you said a Series A, but I didn't hear an amount. I may have missed it. Apologies. And then second, what do you know about the safety so far?
Sure. So we're raising $10 million to get us into humans, to get us through the first in man stage, and that will be all of our IND enabling and manufacturing, GMP manufacturing. And at this stage, the tox in we haven't done anything bigger than a rat, but the tox is basically clean. So if you look at from the theoretical point of view, the cells that you would expect to be most at risk from antioxidant therapy, which is essentially anti-antioxidant therapy, which is essentially what this is, would be in the brain and in the heart. And there are no issues at 25x therapeutic dose in either of these cell types. To the extent that you can, of course, measure them in a rat. You know, the dog will give us finer grained cardiac and some neurological outputs, but we think that there is a very considerable therapeutic index here. So that when we're giving the therapeutic doses, because it's a catalyst, you know, the upside is you don't have to give very much for it to be very highly active.
Terrific. Next question.
Go to Tom, and then we'll go to Michael.
Yeah, Ross. Hi, Ross. Absolutely fascinating. The manufacturing side of things, you know, you've got that nailed in patents, as you say, but you know, the sort of dosing you've given, you know, to humans, what are you predicting?
Well, in cells, IC50s in the sort of tens of nanomolar range. So the actual doses for humans are going to be very small. Obviously, there is a fair amount of PK/PD work that we need to do to work out the exposure. So far, we've identified that the half-life in a rat is about between 22 and 24 hours. So we think scaling up to humans probably going to be about weekly dosage. And it's probably going to be in the order of, you know, 5 milligram dosages that are then going to be completely eliminated. Because of course, one of the potential worries about something like this, that as a catalyst, is that if it accumulates, it'll hang around doing mischief. So so far we've found no evidence at all of accumulation, and this is down to the point of, you know, reducing tissue to plasma and then measuring the number of silver atoms in there. After 3 or 4 half-lives, we found no silver atoms at all. So obviously this continues as we move up the feeding chain, but at the moment we don't have a signal for either accumulation or serious toxicity.
Okay, as far as these reactive oxidative species concerned? You mentioned ROS and sulfate, but evolutionary, there's these reactive nitrogen species as well.
Yes, but this is a very specific stoichiometric relationship between the molecular architecture and superoxide. So this isn't something that will sort of non-specific charged molecules aren't going to be to enter into this. So this is actually quite a specific mitochondrial effect. But that's a very good question, because clearly, if you have something that is a catalyst, you worry about non-specific things, but actually it turns out it is actually surprisingly specific.
Okay.
Fantastic. So last question, I think, from Michael. Where's
Hi,
Michael?
Ross.
Hi.
Good to see you. Really exciting. I just wondered more question around the pipeline side of things with Ross. Puts into play in terms of that pipeline you mentioned. I can see the benefit of going off KRAS and the like there, but what are your thoughts are on the pipeline side of things? Pipeline different therapeutics. I know that you could potentially use this to act as a different modality. What do you mean by pipeline on that?
Yeah, so the molecule itself, we think, has lots of different uses. So specifically, KRAS wild-type tumors like glioblastoma, which are across the blood-brain barrier, we think are a potential sweet spot, because, as you know, the blood-brain barrier is a problem for a lot of therapeutics. So we're interested in primary and secondary cancers in the brain in general. AG5 will also act in concert with therapeutics, and specifically we've been testing with the G12C inhibitors and find that we've got a really interesting potential story of preventing resistance to G12C inhibitors, which is a potential problem. And the third potential area that I think is becoming fashionable again is the radioligand therapy, and of course radioligands kind of died what turned out to be a temporary death when they didn't really work very well because of signal to noise. And what we found is that AG5 will catalyze exogenously generated reactive oxygen species like external beam radiotherapy and also alpha emitter radiation. So we think that it's a way potentially of giving with a radioligand to increase the signal to noise ratio in the tumor.
Fantastic. Now I'm just mindful of time because Graham
is about this all day, of course.
Yeah, Graham is waving at me in a very special way. I think we should wind it up and move on. Graham, would you agree?
Yes, absolutely.
Yeah. Ross,
we're in good time.
Ross, can I just say, firstly, man, fantastic. Anything that is that new has got to be interesting, and thank you for sharing it with us. People can get in touch with you, I'm sure, on LinkedIn or any other of the vehicles, and we look forward to talking to you more. Let's move on to our next person on the XY Factor today, and it's our pleasure to introduce Leon. Now, where is Leon on my screen?
He is here. He's waving.
Where is Leon?
He's got a lovely purple jumper on. He's waving.
Oh my God! There I go. I've got him. He's down the bottom. Leon, welcome. So maybe you could just introduce yourself, tell us who you are, where you are, and what you do.
Yeah, sure. Tony, Graham, thank you very much for the opportunity. Highly appreciate it.
You're welcome.
So I'm Leon Spijkers, Chief Business Officer of Similia Therapeutics, and I joined actually the company a year ago. Similia started in 2017, so I'm not the CEO, but I'm also, joining forces with the team. And I think it's interesting to also explain why the background mentions the European Space Agency before we actually dive into the assets that we're developing. It's actually our background is within the European Space Agency. The ESA actually started several years ago a big consortium. It's called the Melissa Consortium, and it consists of over 60 entities, academia, commercial entities that have more than 100 researchers for year after year working on a very specific topic, interesting for the ESA, And that's actually to enable human astronauts to survive long space missions. You can imagine if you're on a rocket to Mars, you can't bring all your food and water and oxygen with you. It's in terms of volume too much. So you have to go into a circular system. You have to reuse everything that is on board. And that's actually the scope of Melissa. And we are part of this Melissa consortium. And you can imagine that Melissa, in that sense, they focus on two things. That's the circular system, often using microorganisms to allow cleaning waste products and also generating gas like oxygen. And at the same time, human physiology. Of course, that is over a long space flight also endangered in many different ways. So you can imagine that these entities, they generate a lot of IP, some relevant to pharmaceutical applications, some not. And we, as a partner of the consortium, Similia Therapeutics, is actually the valorization partner for all the pharmaceutical related or relevant IP. So we can actually cherry pick. We cherry pick the assets or IP that come out of the Melissa consortium. And we actually, put that in our drug delivery pipeline, or drug development pipeline, I should say, in the end, to hope to bring back, of course, all the IP that is generated and intended for space, also bring it back into use for Earth. So that's Similia Therapeutics as a source of having, new assets in development. As I mentioned, we started several years ago. So we now have a couple of assets, mainly on cardiovascular. So hypercholesterolemia, atherosclerosis, but also NASH, the metabolic disorders. And we're also now carefully looking into CNS. So it's a diverse pipeline. And I have to say that, our first asset against hypercholesterolemia, that is the furthest developed. So, in that sense,
just this thing, I think it's fascinating that you're part of the space solution. So, I mean, are you part of a grand plan, or is this literally you go through the cupboards of this lot and have a look? Because I mean, you've got 3 different classes. I mean, just tell me where they came from and what's the connection? Is it random or is it strategic?
Yeah, it is, of course, from our side, obviously strategic. For the ESA, of course, they have so many activities. But at the same time, it's really good to realize that the ESA actually funds and manages the Melissa Foundation for many years, and they will also do that many upcoming years. So this is really part of a ESA focus that is long lasting. So this is not just, a byproduct of any activity. No, the ESA is quite serious on the Melissa Foundation. They invest quite a lot of resources in this. And Similia being one of the partners of this foundation, of course, in that sense also makes a part of a bigger plan also for the ESA.
And are you a selling organization, or do you take the asset at a certain phase and flip it in?
Yeah, so the Melissa Consortium, of course, has a very focused output. And that's not primarily developing pharmaceutical products. This is really something that our unique position is also within the Melissa Foundation, because we have access to the IP, but we don't have to, of course, incorporate it. We really can cherry pick, but at the same time, this is our cup of tea. So the ESA will not fund all the pharmaceutical drug development programs that we initiate based on the IP of Melissa. It's really, in that sense, a private company.
So in a way, we could think of the ESA as a university with and you've got a pipeline agreement with the university to bring in assets that you want. Is that a pipeline agreement forever? Is it a pipeline agreement for a period of time? And then I suppose the question is ESA is a government-funded foundation, is it? So are you, what's your goal? Are you going to you're going to float? Are you going to, what's your goal? Because it looks like you've got a load of good stuff.
Yes. So, the ASA will, as I mentioned before, will not fund our programs. We do this on our own. What we actually have as our own business model, and this is really the Similia Therapeutics business model, is actually cherry pick IP that is eligible for the pipeline, for the development. We actually raise funding to make sure that we can reach, the clinical stages with the assets. And in the end, it will be a licensing deal with a pharmaceutical company, because we will not approach the market ourselves with products. We really develop it into something that gains value for pharmaceutical companies. So it's at the moment all still preclinical. We have for 2 assets also a proof of concept in humans, but this is a preliminary proof of concept. We are still in that sense a preclinical company with preclinical assets. But we are currently raising a 10 million Series A to move forward to a clinic with at least 1 asset, and also expand the preclinical data on other assets. And we're also very much open to a bridging funding or a pre-Series A of 500K to actually, pinpoint the API and MOA of a specific asset that we want to develop further.
I mean, this is quite a well-trodden model, really. That you, as I say, have a pipeline agreement, you have some seed funding, and then you have a Series A. I mean, have you got? So are there any investors involved at the moment, or is it very much you're looking for your first set of investors?
Yeah, so of course the ASA gave us some starting funding, the seed finance, but we also have private investors on board already. So we raised already a couple of million via private investors, and in that sense, it's now time also to move towards a Series A, either with a bridging finance or just a full Series A.
And I mean,
that would actually.
You sound like a sort of mature company, but not quite in your financing. Is that how you would describe yourself?
Well, in that sense, we're still preclinical, and on one side, it's very attractive to have a big source of new IP, but at the same time, you have to have, of course, also the capital and also the resources in terms of human capital to make sure that that is integrated in a drug development pipeline quite efficiently. So at the moment, we have already quite some assets. I think it's up to us to actually expand the value of those assets already.
Would somebody take a pipeline? Would you allow an investor to take part of the pipeline agreement? So in other words, a bit like the IP group in London that put shell money into something, and then you effectively own the pipeline. So you own the first dibs on what comes out of ESA, which is very attractive.
Mm-hmm. Yeah, well, in that sense, if there are interested parties to also, have a saying about certain IP that can be, commercialized, that's something that we can discuss with. But at the same time, we don't have, the full rights to claim all the IP. We have access to the IP. So in that sense, it is not said that all the IP generated by the Melissa Consortium is ours. That's very important to state that. We have a first right of refusal, I would say, to the IP.
Right, interesting. Graham, how are we doing for timing? Hello, Graham?
Yes, we're doing good.
I'm going to open it up for questions from Tom, Christine and Michael. So maybe
absolutely.
Christine, you're waving at me as...
Yeah, I fascinating. I was not prepared to go into space this morning.
You should have come really with us on the psychedelics earlier last week. But
yeah,
anyway.
I mean, I need more coffee for that.
Yeah.
So as you have what I assume is a pretty broad set of IP being generated as you think about the kinds of potential things that are being worked on within this consortium, I'm just curious as to why you've chosen to go into initially cardiovascular metabolism. Was this something that you just saw as new and novel? Is this an area that you have passion for? Just want to try to understand, given what I'm sure is a lot of different things, why CVM?
Yeah, I mean, this is part of the chronology of having, you know, access to the IP and discussing the IP, what is relevant. This was just the first asset that actually, you know, was of interest to us, and in that sense, that's you know, something has to be the first. So in that sense, this it's a microorganism that's the source of you know, the APIs. And yeah, we saw a lot of benefits in this because of the strong cholesterol lowering capabilities. Having said that, also, you know, other disease areas could benefit from this. And this is the furthest developed asset that we have at the moment. We also had a proof of concept in humans, and quite an interesting preclinical package. We tested these compounds in many different species, mice, rats, pigs, hens, and they all gave a very profound LDL lowering effect. So in that sense, that gave us a very good basis to find a new kind of statin alternative that is highly efficacious and at the same time also highly safe, because there were no red flags on any time and any occasion on safety issues. Also in the human study, where you had the patient self-reporting, nothing stated there. So in terms of safety, we have very good hopes on this asset, and also in terms of efficacy. And this is just, you know.
And is this then, you've determined what the compound is that's being produced by the bacteria and you're synthetically making that, or are you having the bacteria make it for you and you're recovering it that way, or are you giving the bacteria?
Yeah, well, if you follow the patent lineage that we developed already on this part, you can see that we actually are zooming or narrowing into the API that is responsible for this. And this is always, you know, a lot of, you know, research and development to try to find that needle in a haystack. You have a big pool of compounds. Which one is the active ingredient? And if you follow the patent lineage that we have, you can see that we can continuously zoom into the compounds that are yeah, relevant.
The actual patent estate, who is maintaining that estate? Is it ESA?
No, this is really, in that sense, we are on the one side very spoiled to be in this consortium, but at the same time, that does not mean that, you know, we have unlimited resources. This is our cup of tea. We do this, so it's also our IP, our patents, and our patent costs.
But this must be a very costly, you know, endeavor for you, because you seem to have so many IP, a large IP estate in a large basket.
No, because the IP is from, this is Melissa IP, and they maintain, of course, the IP status. The moment we say, after cherry picking, we are interested, then it becomes our IP. But it's not that we have to, you know, take care of the cost of all the new IP that is generated. That is, of course, not a fair deal also for us. You know, as a company, we can say, okay, the moment we are interested, we take over the IP, and then, of course, that becomes, you know, our cost as well. So it is limited to the assets in development.
Mm.
Michael.
Yeah, thanks, Leon. Really interesting. I just had a couple of questions. I assume these are, given that it's come from ESA, these are natural products?
Most of them are. Some are synthetic because, as I mentioned before, there are tens of universities involved as well. So medicinal chemistry departments, you know, can also provide with a very interesting assets. But many of them are indeed natural sources. Yes.
And is you know, part of what you're doing with your company is to you know, find ways, let's say, for the natural sources to you know, grow up the bacteria and the like, because I know that's a you know, big bottleneck in this kind of space.
Yeah, it's always part of our activities to also make sure that we have upscaling protocols, etc., also developed. So for this microorganism, we also have an external partner that actually developed for us the upscaling protocols. we own the IP on this as well. So in terms of, you know, use and in terms of manufacturing, we own the IP. And in terms of that, say, I don't know if it's right first refusal or
Michael, I think we've got to cut it there. Sorry about that.
Well, we could have that. Finish off that question. Finish off that question.
We'll finish off that question, and then we'll move on. Thank you, Michael.
Yeah, no, it is more around sort of licensing play. So that's potentially, if you can cherry pick, and let's say you can cherry pick natural products, and you find ways to scale that up, and potentially there could be some really interesting, unique compounds, not 2D based, that could be, you know, beneficial to, you know, compound libraries and screens for, you know, pharma around the world. So I just wondered if you guys looked into that.
Yeah, it's true. No, we haven't actively looked into that. You know, it's mainly based on focus. We of course have to focus, and there are many different ways to commercialize, you know, assets. And but we chose to have actually, you know, the pharmaceutical approach. If there are companies that are interested to make, you know, a Prestwich 2.0 database or something like that, or compound based, it's fine, but it's not our primary focus.
Great.
Fantastic. I think that we need to thank you, Leon, in a very big way. Leon, if anyone wants to contact Leon, please look at LinkedIn or email or send a carrier pigeon if you're old fashioned. And we're off to the next one because we need to keep on track. So thanks again. Now, the next one we're going to introduce is Peck. So Peck, let me try and find you on my screen.
Remember the oranges.
Where are you, Peck? Where are you? California. There you are, Peck. Peck, welcome to
Hi,
Peck.
Danny.
Hi, lovely to see you.
Graham. Yep.
Maybe you could introduce yourself to the gang and say where you are, what you do, and who you are.
Great, thank you guys. I am Peck Lum. I'm the co-founder and CEO of a AI-driven biotech company called Arjuna. As you can see from my my lemon tree, I'm actually based in the Bay Area, so it's a bit early for me. So sorry, my voice is a little bit weird today. And we have so my background is in genetics and genomics, and really happy to be here today.
Terrific. And so let's start with your company. So what's it do?
What does it do? So, you know, it's there are like biotech companies, and there is this phrase where people call it the tech bio company, and we think of ourselves as a technology company, but you know, with a killer app, which is drug discovery, a very difficult and complex industry. So we are a computationally driven, data-driven biotechnology company. When I say tech bio, I really mean that we have built a computational engine, what we call the SmartA engine, that drives drug discovery from the get-go. We use our algorithms to look at patient heterogeneity, and that is really what we really focus on. And after studying heterogeneity, our engine basically predicts compounds that that are deemed efficacious, for that. We use gene expression data, molecular data, to study the biology, and we have built a great pipeline in this. A few years, 5 years we've been in business. We have built a great pipeline. We have a liver cancer drug that is ready to go to the clinic. That is in manufacturing process right now. We have a heart protection drug. Can
you just Peck before we go on?
Sure.
So you're a tech bio company, so you've got this piece of mathematics that can skim through data, and it can lift up things and make them flash and say, "Pick me, pick me." Well, that's because you've got lemons behind you. What is it you got, and what data are you trawling?
Yeah, we are very into RNA data, gene expression data. So we've got we look at gene expression data in human disease tissues. We are very focused on oncology, but we also do all kinds of different diseases. The engine is disease agnostic, so we've had kind of our hands on, autoimmune diseases, skin diseases, brain diseases, but, in terms of drug development, our company is focused on cancer and cancer care. So we have data that we basically take from the public domain. We also have private data, but mostly public domain data where we use gene expression that is generated by many different people on, from various parts of the world, of course, various cohorts. And we like that because we study heterogeneity. It represents heterogeneity that we think, should be represented, say, in clinical trials, or when in the human population. And we have algorithms that really look at all these very dirty, noisy, heterogeneous data and look for the subtypes of diseases and for I would say recurrent biological signals before we set on to predict compounds, again, gene expression from compounds that will allow us to identify which compounds whose gene expression, I would say, quote unquote, reverse the disease biology in silico.
Is this huge sets of data? I mean, is this like awesomely large sums of data or not?
I would say it's awesomely heterogeneous. Our engine can deal with large data sets or small data sets as long as we feel like the heterogeneity is represented. We have done, for example, for tumors, for cancers, there are a lot of I would say disease tissues, gene expression profiles available. So we have thousands of those. Sometimes we look at single cell data, and those could be tens of thousands and of single cells. And sometimes we look at more rare diseases, and those could be in the hundreds. And so we, it really runs the whole range, and the engine could look at that and figure out if there is heterogeneity. Maybe some diseases, like more rare diseases, are not very heterogeneous, and that's okay too. But we want to go in knowing how heterogeneous the data is and whether we can find recurrent biological signals.
So, I mean, we're seeing there's a lot of people who are in this tech bio space. What's in your magic box? What if I was to give you a box and I ask you to open the lid, and what's special in your magic box? Right.
As when we first started, I think, the concept of really AI-driven drug discovery, was kind of an alien concept especially for investors. Is either you make a software or you are a biotech company. But, for us, we wanted to build a platform that could generate a lot of these shots at goal, good shots at goal. So our magic is that we study biology. So a lot of the other magic of other companies, they might, have, their sweet spot in doing chemistry, and with their docking chemical compounds onto targets of interest, or they might study images. They might study, natural language processing, where they screen literature. But at Arjuna, I think our uniqueness is we are really into studying biology. How do we actually discover subtypes, patient subtypes, which we think is super important for drug discovery by using AI and computation? So that's really kind of where we start from. And that's a really difficult endeavor because, as you can tell, first you have to be able to handle gene expression data that is from various sources. But like I said, we like that kind of heterogeneity because our algorithms are made to really detect recurrent signals in biology. So we start from biology, we start from human disease biology, thinking that, that is a really good way to start if you want to understand why things failed in phase 2.
You're big, so just because of time, so you've got a magic box, and in that box you've got some tech bio. So where will the value inflections come? Where are they in front of you?
I think the value inflection comes from really, the very same thing as, as what, the value inflections are in a drug discovery, any drug discovery program, right? The the more you bring it, across the finish line, the better the value inflection is. So, our engine can now process data from end to end, from ingesting large amounts of data to outputting subtypes, patient biomarkers, and preclinical models actually that matches it, and also compounds, drugs that I think will be a good one for us to start with, all in 24 hours. But the value inflection point really comes when we take that and put that in the lab. And for us, we don't have to test tens of thousands or millions of compounds. We can test, we've shown for many programs now, we can test fewer than 30 and be able to get a few that has what we call development legs, and be able to bring those further and further forward. So like I said, we've got one that is ready to go into phase 1. We have a liver cancer drug, a novel molecule. We've got another one that is the a heart protection drug that we want to use as a heart-safe chemotherapy against anthracyclines. That already has been out licensed to a Chinese pharma company, and that's undergoing formulation. So you can see that it's really, really fast.
And are these reprofiled drugs, or are they NCEs? So
they're all NCEs, but they can be could be reprofiled drugs. So we have in our library a starting library of any compounds that has gene expression in it. Those could be Merck's compound and anybody else's compound. So they can be both.
And last question, and then we're going to pass the panel.
Yes, I see Graham doing this. Yeah.
Are you looking for funding?
Yes, we are. We are currently raising a B round right now, and anybody who's interested, please let us know.
What size of round is it?
It's about we're planning to raise $30 million, 30.
Fantastic.
Well, we got a question from Jai actually for this. Fantastic. One. I know you've got a tech head on, don't you, Jai?
Yeah. Well, it's actually biohead. So my question is, how do you decide which indication or therapeutic area to go after? Because I'm looking at your website, it looks like they are quite a lot.
Yes. Yeah. So we have, as any platform company, the engine can ingest a lot of things. So we can see that the engine is disease agnostic, but then when we actually bring that into the wet lab, we actually have half our team are veteran drug developers. We take a look at many things, including in the unmet medical need, market size, maybe market size is not as big, but unmet medical needs are a big thing. Whether we have the compounds that we find, whether it could go as it is, or whether we need to make an analog from it. So we just put on our drug development hat on after that. So if you look at the things that we advance to IND enabling, they are mostly in cancer, and the rest we actually partner off. We have partnering on various things that we will not bring in ourselves because maybe of lack of expertise or talent at this point, or that we think we should focus on other things. So we are really focusing on cancer and oncology. Is what Arjuna itself does, where we bring things into the wet bench, into IND enabling studies, and the rest we've been partnering out just because we have, like my Maya lemons we have a lot. So we
Okay, Christine. Christine's got a question.
I do. So very cool, and obviously, as we think more about how do we do better in drug discovery, this is an important part. Taking the AI and all this data that we've accumulated. But my question is as you're drawing, especially the data from public domains, how do you decide, or is this part of the programming, what's actually valuable? Garbage in, garbage out. So how do you know that it's not garbage in and that you're getting really important pieces of information to really accumulate and run through the tech?
Great question. We have looked at data, and one of the things that we start off with is that because we don't know subtypes, nobody can be the expert and say, hey, this is my subtype. So the engine, so all this is part of the engine. The engine actually will be able to discover anything that it wants in terms of subtypes, And then, like you said, sometimes the subtypes could be garbage, like the robot dropped the plate or something. We don't know, and the engine doesn't know. However, what it does is that is an approach of ours, is our secret sauce, where we take these very heterogeneous data for that same purpose, as you've said. But then we look for recurrent biological signal. It learns what is recurrent. And then the idea is that if it's recurrent, then the chances of a subtype being garbage is extremely, extremely low. So it works on that premise. So we're not a company where we have testing and or training. And because that part we believe cannot be trained, because there is no labels, there's no ground truth in terms of gene expression and subtypes yet. So it learns, it basically discovers that, and then it learns the recurrent signal. So it is part of our programming.
Great. Question from Tom.
Yeah. Hi, Peck.
Hey, Tom.
When I think of secret sources and, key, tests, in days gone by, the chemistry, the med chem, use sort of the Lipinski law of 5, to get you to a druggable, target, or in your case, a drug with legs. Of all the criteria you use in this engine, what would you say are the weakest points?
I guess, our weakest point would be, first I'll tell you what my strongest point is that, we have a drug database that we have collected probably now over 500,000 of gene expression profiles of compounds. And these compounds could be any compounds, could be Merck's compound, could be a tool compound, could be natural compound, all kinds of compounds. We use that as our starting library. So that's a scaffold that we actually use. And then, your mentor once said, every good new drug comes from an old drug, right? So we like to use that. So we believe that, of over 500,000 gene expression profiles and 22,000 unique compounds, that is actually, one of our strongest points, where we start from where the engine shines the light on. I guess one of the weakest point of the engine, if I have to say it, is that, I love rare diseases, and rare diseases are actually hard to come by in terms of gene expression of their disease tissues, in the public domain. So I would say that if we could go after rare diseases, very rare diseases, for example, we might not be able to get to that quite easily, and we would have to, do collaborations with people. But having said that, for example, we are looking at a rare disease, a childhood cancer disease called diffuse DIPG, diffuse, something pontine, and it is a really bad disease. But there is enough smaller data sets, but multiple of them, that allowed us to actually now look at drugs that might be useful for that. So I guess the data of disease tissues might be, something, but we feel like that's an important thing, that if we can't find the right disease tissues, we shouldn't be doing it in the first place.
Okay, Michael, you got the final question there.
Then we need to move on, Graham.
I'll be very quick.
Yeah,
like,
Michael.
really exciting. Using gene expression levels of people who've been treated with compounds, and my general question generally is how I like that concept, and it sounds differentiated from Crystal Pi and many others out there, that taking a sort of computational, more physics-based approach. But I just wonder, looking at your the website and the profiles, it seems that for certain cancers you've like raced ahead, for some others it's early. And I had a general question around from wanting to go after a certain cancer indication, let's say, but I see cardioprotection's also quite high, and you've raced ahead there. Do you just press go on, say, breast cancer, and the engine just goes to work, or are there more subtleties, more nuance that means it's harder to progress with some indications than others?
I love that. We actually now, because it takes the engine in 24 hours to do what it takes. When we first started, we were much clunkier. It took us probably a month to plow through everything. But now it's the engine has always been on AWS, on the cloud. And we have, so right now, for example, if somebody comes to us and say, help us with this, or even COVID-19, actually, we have collaborations with EDDC of Singapore and with a Korean, biotech company. It was the moment we got data, to the moment we could test the drugs, probably it's only about a week or so. So right now, what we do is like, people come to us, or we sit down, every week, and we say, this should be quite an interesting new thing for the engine to ingest. So what it does is that we give the engine, diseases, and it trawls the databases, with our data scientists. And then once we find data, literally we push the button and everything is then outputted, in about 24 hours. And then as a company, we sit down and say, okay, we just finished head and neck cancer, for example. We think and then we start to look at the market of head and neck, the unmet medical need, and so on. And we said, this might be very interesting for us. We've got subtypes that looks interesting. Let's just now start to test it in vitro, then in vivo. So that's how we make decisions on that, on what to go after, really, because,
I'm going to have to cut you there, Peck.
Yeah,
thank you very much. That was wonderful. Thank you very much.
Fantastic. So let me just thank Peck for being awesome as always. Thank you very much, my friend. And then we're going to go on to our last one of the afternoon.
From one AI to another AI.
In our XY competition, which is our inaugural XY competition, I'm going to welcome our friend Murat. And Murat, maybe you could tell everyone who you are, what you do, and where you are.
Definitely. My name is Murat Sunavoğlu. I'm a CEO of Antiverse. Antiverse is an AI biotech company planning to change the future, how these drugs are discovered. I'm originally from Turkey, but I traveled around quite a bit before I settled in UK. But today, because of COVID and other related issues, I'm calling from Istanbul.
Great. Okay.
Fantastic. Well, welcome. I hope the weather's better than it is here. But anyway, welcome. And maybe you could just tell us what's the company do? What's your technology?
Definitely. So as you already know, there are many difficult targets associated with cancer, heart diseases, lung diseases, and unfortunately, only less than 1% of these difficult targets have been explored. And the reason is it's very hard to extract them and work with them. Antiverse is finding an alternative way, using the blueprints of those difficult targets, amino acid sequences, to engineer or design new antibodies against them in silico, and to do that in parallel in a very short frame of time. So that's our big vision. It is introducing hundreds of new ways to treat cancer. And to be able to do that, we are building a platform. However, that's a big goal. So before getting there, we had to invent something else, offer some short-term value to pharma and biotech, and that is identifying diverse and rare antibodies. It's kind of a process improvement to a traditional screen.
I mean, are these, so you got the, if you like, the book of traditional cancer targets. So you're looking for new targets, and how are you finding them?
Right. So we are building the system so that it can work pretty much with any GPCR and ion channel from a technical perspective. But of course, we are focusing on to a few member families. The way how we approach, it's not our expertise to select the targets. So we work with some experts on a consultancy basis. They craft the long list. We take the list, take it to pharma, say, we have only enough capacity to do maybe 6 of them this year. You know, which ones we should focus. And they say, oh, these 3, if you could solve them or find an agonist or antagonistic antibody, that would mean a lot to us. And that's how we kind of prioritize that work.
Is that looking at sequences of the antibody and looking at docking? I mean, is that the sort of idea?
Yes. So we are Antiverse, the name of the company is coming from this antibody universe, right? The whole universe of antibodies, maybe 10 to the power of 30. And to be able to pick the right antibody for any given antigen based on its epitope requires a lot of work. So what we decided to generate a very large data set of antigen sequences and antibody sequences, then infer that relationship with mathematical models. And the next times, once we get a new antigen, we basically study its epitope. And then for that epitope, we generate new antibodies and then test them in the lab.
And how does that fit in with like tri-specific antibodies that we're now looking at, and T-cell engagers? I mean, we're moving into a new world, really, where we've got many moving parts, which I find fascinating. I mean, how does all that fit together?
We work in a scaffold agnostic way. We design the paratopes, if you will, and then it is up to the partner company to either bring into a bispecific or a tri-specific.
Is your goal, Amirat, to be a IP company or a service provider or a mixed model, or what's your business model?
Right now it's fee for service because we want to do as many projects as possible and then in return collect data, offer some value. But in the long run, it's licensing. So the way how it works, you keep the platform independent and set up joint ventures around a group of licensed targets. If you look at F-Star from UK or Atomwise from US or Nimbus. So we will keep the platform independent and set up collaborations around the targets.
I mean, how much are you looking for and what's your value inflection?
So we recently closed a round. Since 2017 in total, we raised about £2 million. Our inflection point is showing 12 months from now that we can actually engineer antibodies against 6 difficult targets, of which we already picked. And for the next 3, we are talking to different pharma. And how much we are going to raise, it's a little bit not clear yet, but probably over 10 million, between 10 and 15.
And will they pay you just for fee for service, or will you get royalties?
What we will show before the series is the licensing agreement signed. So it will be milestones and royalties. Because the targets we are working with haven't been cracked yet. So the value we offer is suddenly being the first-in-class therapeutics, and for that, we think that we have a very good chance of negotiating a licensing agreement with milestones and royalties.
Good. And I mean, if you were to say a couple of words to investors about why they should invest in you, what would you say?
Well, we are definitely looking for investors that buys into the big vision of AI and AI's impact in drug discovery. We want to keep the company independent so that it reaches its full potential, full scale. And I know it's not for everyone, but we definitely want to pick partners that would see this company reach its full potential in the next 5 years.
Fantastic. Graham, I'm going to ask for questions now from our...
Yes, be great. Would you like to go first, Christine?
Sure. So, you know, I think it's very interesting trying to understand either new targets or new ways to address those targets in various antibody formats is really important and fascinating. I guess kind of what's the status so far of your collaborations, and just trying to understand sort of, have you proven out the platform from a proof of concept point of view? You know, have you gotten into animal models either yourself or with your collaborations? You know, have you done multiple different types of formats? Just trying to understand kind of where you are in the proving that this is a platform and it's delivering.
Yeah. So what we have done so far is 3 different proof of concepts. First of all, we are post-revenue, so we offer some service, generate some money, work with pharma and biotech companies. What we do so far is working with an antigen and generating new antibodies, or doing the screening with phage display or immunization and looking that whole repertoire and then picking diverse and rare antibodies from there. And for the recovery part, for selecting the diverse antibodies, we recently completed a project, identified 238 unique and very diverse binders, and 92% of them turn out to be binder. And also we generated new antibodies that wasn't part of the library at all. And it's been shown that they are also binders. So quite proud about that.
Excellent.
Right. Yeah, you're on mute, Tom.
Okay. Yeah, absolutely fascinating. You know, my concern always comes back to the intellectual property with regard to how this is driven, your particular library of either rare or not so rare, antibodies. How are you protecting yourself?
Yes, I think the biggest IP we have is, there are two things. One of them is the machine learning models, and advice we got from the UK offices are that patenting the software and protecting, enforcing it is very difficult. So we've been advised against it. But they said, oh, you definitely have a lot of know-how about the data generation, validation, the high throughput methods, and so on. So maybe you can at least protect those optimizations of certain assays. So that's something we are looking into. But the real IP is the generated assets coming out of the platform. And that will be, so far that belongs to the customer, but going forward, that will be the joint ownership with partners.
Great. Michael.
Michael.
Hi, Mira. See you again. Just a general question in terms of how fast now can you, from choosing or selecting a, target to go after, how quickly do you believe, or have you been able to model, can you generate antibodies to a target?
It is not fully automated. So the fastest we have done so far, historically, is a month. But I think the real bottleneck is how many of them you can test, really, right? For the efficacy purposes, for example, we can find thousands of binders in certain cases, but that's not necessarily, the expressing purification and an ELISA and SPR and so on. It's not possible. So what we are trying to do right now is kind of prioritize that list in a certain way, that it can select for function. So far, it only selects for binding. It only selects for affinity, but we want to select for function as well.
That would be based on how much information you sort of pull in your wet lab evidence that you'll then put back into the algorithm, and it will be able to start to predict, in effect, what sort of bind you'll get and what assumed signaling effect you'll get on the back end.
Correct. Once we have an antigen, what we do, we predict the epitopes, right? And there's literature out there for certain difficult targets, or pharmaceutical partners already have some positive controls in terms of small peptides, small molecules, and so on. So based on that information, we can design antibodies that only fits onto those epitopes, right? Yeah. It doesn't say that it will have the same efficacy. At least it will reduce the search space or the number of tests you have to do.
Just a cheeky last one, off the back of what Tom mentioned. I just wondered if you'd, spoken to regulators and whether they're open. We're getting more and more, as we just heard from Peck, increasingly where, informatics computation can potentially, revolutionize the way we, identify compounds or agents to go after targets. And there'll be that increasing point where, where's the novelty, where's that intellectual, input. I'm just wondering if, you can get the computer to do some to a certain point, but then you need that ingenuity, you need that wet lab work, and how much could there be a balance so that you can start to, file IP on, things that have been spat out from computers, in effect?
How far you can push things, right? Well, you see, there are a lot of companies working in the small molecule space, right? Because it's a little bit easier to model, the interactions that are better studied and structures are more readily available. For the biologic space, I've seen companies all the way from target identification to developability improvements. So I've seen, affinity maturation, developability optimization, solubility optimizations, and so on. But I think that's where the kind of vision ends right now. But maybe in the future, it will be all the way to preclinical studies and so on.
Great. I think we're going to round up there, Tony.
I think we're coming to the end. Murat, fantastic, man. I love all that antibody stuff, and we look forward to you cracking through and doing some great stuff. So thank you very much. That's the end of today's XY Factor, and I think it's a great start. Thank you to our panel, to Tom, to Catherine, to Michael, and our guest appearance as well of Zhizhang, and yes, and then we've got all of our people who joined us: Jeanette, Ross, Leon, Peck, Murat. We've had a fantastic time. Please come again on Thursday, and please come on Friday when our friend Katere, I think, will announce the winners and the order of winners, which will be a great pleasure. For today, we've got one more gig at 6 o'clock, which is what, Graham?
We're going to be interviewing the managing director from Fidelta. So that would be a very interesting way. And then we'll do the mix up as well, so that we have some networking in that.
Start tomorrow at 9 o'clock with Michelle, I think, is speaking from MSD.
That's right.
As an update on what's MSD looking for. So please come to that, because you'll get some signals about what they're after and how they're getting on. And we look forward to seeing you for the rest of the week. So thanks again to everybody. Take care. Have a safe trip, and bye for now. Ciao, ciao.
Bye.
Thanks, everybody. Bye-bye.
Thank you. Bye-bye.
Bye.